Thank you, Professor Chan. You didn't use all the time, so I thank you. The next presenter will be Dr. Klaus Urbahns. He is a senior VP and head of discovery and development technologies at Merck. Dr. Urbahns holds a PhD in chemistry and has extensive 25-year experience in the pharma industry.
Thanks, Dr. Opper. Michel, Claire, dear colleagues, dear fellow scientists, ladies and gentlemen, it's a particular pleasure for me to be here today. It's humbling, and I feel deeply honored that I'm allowed to speak here on the occasion of Professor Michel Revel's 80th birthday. Thank you very much for having me. And you might well ask, what's that guy doing here? Merck, who's that actually? And why would you actually want to invite someone from that company? Well, I can briefly introduce the company, if I may, for a moment, and then explain to you a little bit what links us to Michel and why we are why we have become so great friends, I think, over the years. This company is actually has something with Michel in common. It is also celebrating an anniversary this year. It's 350 years old, and thereby it's actually the oldest pharmaceutical company in the world. There's a lot of confusion with our name. Merck is actually a family name, from a family, and unfortunately, after the First World War, the American government decided to actually take all our American laboratories and build an own company from that. And ever since, they are called Merck in the United States and in Canada, the blue part here, and in the rest of the world, we are called Merck. It's 50,000 employees, and we have 3 branches, actually. I want to briefly go through those. I told you already about the story that we are 350 years old. One particular event where the link to Michel is coming from is actually happened in 2007, where we did one smart thing. We acquired the company Serono, and that was actually a good thing for us, and you will soon find out why that was. The whole story started really by someone called Friedrich Jakob Merck, who bought a pharmacy. Pharmacy is really at the core of the company Merck. But it's also fair to say that we have some other businesses we are in. One, for instance, you probably are already a proud owner of a Merck product, which is in your cell phone. All cell phone displays, or most of them, certainly all the Apple ones, contain displays from Merck. Actually, Merck scientists invented that and made flat screen technology actually possible through that invention. They're also very strong in chemicals for electronics. If you want to create new chips, then that's something we do. And another thing is anything that glitters, actually, we are good at that as well, whether it's makeup for the skin or for cars. Anything that glitters comes from a mineral that is called vermiculite, and that we can transform into those kind of colors. Another area that we are working on is life science. It's built out of 3 areas: genome editing, food safety, and biologics. And we are trying to make the life of our fellow scientists easier. That is another area. But most importantly, I think we have our healthcare group. And you see here our pipeline. That's always how you introduce a company to other people. You see 2 compounds here which are dedicated to multiple sclerosis. One is called evobrutinib, and the other one is called cladribine tablets. And I'll briefly touch on those as well. But of course, we are also here for one specific molecule, and that is interferon-beta, which is for the first time being made in a recombinant way, and that was the work of Professor Michel Revel. For those of you who are not scientists, this is how such a molecule would look like. So these springs here and these noodles, this is actually the protein part. And then these little balls here, that is sugar that is associated with that. Trying to make that in the laboratory chemically is today impossible, but you can do it biochemically, and you can try to isolate it actually also from natural sources if you wanted to. What do these molecules do? Professor Chan has already illustrated that a little bit. Interferons or cytokines, small signaling molecules which are released by immune cells and actually inform other cells that inflammation is going on in the body somehow. And there are two types, as has been already alluded to. Type 1 interferon being interferon alpha and interferon beta, and type 2 is interferon gamma. And viruses trigger the rapid production of interferon beta. That was very well known. So, there was a product once upon a time which was extremely dirty, very hard to make, and it was called Frone. You can see here still the name, Serono. Every protein that you want to make, actually, you have to have a so-called upstream process where you actually create it, and then a downstream process where you actually purify it. That's a very important distinction. And the upstream process of making Frone was very peculiar. I don't really need to explain it here in this audience in Jerusalem, but you can imagine how this was when you wanted to create major amounts of beta-ferron. So it was best isolated from fibroblasts, isolated from human foreskin as a result of the circumcision procedure. And you can imagine that somewhere in the background there was probably a Serono or an Interfarm researcher trying to actually, with an ice cube and trying to collect the result of this procedure. And this was, of course, not very efficient, as you can imagine. The medication was actually only available in 7 countries and then disappeared completely. But I just wanted to mention that there was already something. It was very low quality, very low doses, because hardly anything was available, really. In came Professor Michel Revel, who made a seminal discovery and really made something which really made a difference. He used Chinese hamster ovary cells, so mammalian cells, and transfected them with the gene of interferon beta. These technologies started to emerge at that time. And in his seminal work in DNA 1984, he and his colleagues published a paper called Effective Constitutive Production of Human Fibroblast Interferon Beta by Hamster Cells Transformed with the interferon beta gene fused to an SV40 early promoter. This is where a lot started. Because for the first time, Michel and his group were able to create interferon beta 1 in really great purity. I mean, you have to, I don't know exactly what this lane 4 was. This is actually the band of a gel, and this is where you would expect this little band here. That is interferon beta 1. And this is where you would expect interferon beta 1 to pop up. And this probably where hardly any interferon beta is in, and loads of other proteins were, and that's probably Frone. And this was the new medication, a completely new thing, absolutely pure and completely identical to the human material. This actually went through the press, and people realized that this was a major thing. A single cell was able to make 300,000 molecules of interferon beta per hour. And it carried on to do so. Fibroblasts, after being activated at some stage, ran out of steam. But these molecules, these cells, they were actually producing the material. It was identical in size, activity, and immunospecificity to the native human interferon beta 1 glycoprotein. Note about this immunospecificity. I told you about these sugar residues to the left and to the right of interferon beta. The fact that they were in place in the correct way really played towards the good properties of this medication. Right. Many people started to become interested in this. And there were many labs visits. You can obviously see Michel here. And I hear that this is Ephraim Katzir, the 4th president of Israel, and also he was head, obviously, of the biophysics department in Weizmann. And this is a 7-liter bioreactor where interferon beta 1 could be made. And people were very proud of that. You can see that. And I think this is a microphone here, so the press was also there. So once Michel Revel's team has actually learned how to make this molecule, it was all about refining this. And the way how you do this at those days, and actually machines don't look much different today, you really look into all the parameters that go into this. So it's the temperature, it's the media, it's the gas you put in, it's how quickly the stirrer is spinning in the tube, and this is what these machines actually did. And then when you really know how to do this, you can really scale up. This is a warehouse where they actually had major amounts of interferon-beta made. This is all a couple of years ago, of course. The company was growing. There was a collaboration between the Weizmann Group and Interpharm, and Professor Revel obviously became the CSO and the leader of that company. If you entered this building here, at the beginning, the company was only on the first floor, I was told, and if you would turn to the left, you would have the production site, where, for instance, the 75-liter fermenters were, and there were also some other larger vessels as well. The 75-liter vessels was actually the process that is even today used for the FDA-registered material. On the right-hand side, you had people. There was a warehouse. There was a production of the media, and of course, there were also all the people you need to actually create the drug. So, all in all, Interpharm was housing 300 people. And of that, there were 70 roughly in R&D. And there were, so it shows there was from a very early step on, it was not just about that one product company, not a one-trick pony show, but it was, they wanted to do more. And there were additional projects that were going on. One was, for instance, about TNF-alpha that people wanted to produce in Escherichia coli at the time. That went very far. And there were a couple of mentionings of Professor Revel in the press, like this one here. And there was another one, another important project, which was around interleukin-6. A very important interleukin, very complex biology. And again, Michel, with his methods, was able to create major amounts of that and make this amenable, really, to scientific investigation. Right. Let me just briefly recapitulate where we are in this voyage of Rebif. It started almost 40 years ago. So 1948, I mentioned the paper which was so seminal. Obviously, there was a lot of work that was ongoing before in order to really ensure that this kind of work was possible. 2 years after that paper, they were having the first production in 3-liter spinners, and then Rebif entered phase 1. Then actually 2 years after that, there was the achievement of producing the material in 5-liter bioreactors, which was actually preparing Rebif for phase 2 studies. And as Professor Chan already pointed out, it isn't exactly clear how interferon-beta 1 actually works. And at the time, it wasn't really clear which indications to use. So what was started then were actually 15 studies in 13 indications, one of them being multiple sclerosis. But probably Michel already had some good guesses whether multiple sclerosis would be the winner. But indeed, that turned out to be the case. In the meantime, the production people were busy and scaled further up to 30-liter bioreactors. And in 1994, Rebif entered phase 3 studies. This is in 1990, the same year where actually the process for the FDA was basically ready, at least at the same scale. And then the company Serono acquired Interpharm, which actually was, of course, a major event for that compound, because Serono really was interested. This was really something which was very, very promising. If you looked into this, 1 year later, there was a European approval, so they knew already that would be coming. 4 years later, there was the FDA approval. And in 2006, actually, Merck did this one smart thing to really acquire Serono and thereby acquiring this compound. There was a lot of further drug development later on. Professor Chan already mentioned all these studies where actually there was investigations after several years of treatment of Rebif, and really the compound came out as a very safe opportunity for multiple sclerosis patients. What was maybe important to say was at the beginning, multiple sclerosis, before the Rebif time, multiple sclerosis was actually called an orphan indication, which means there's some few patients and, well, we can't really much do about this. It was underdiagnosed because medics would hesitate to give the diagnosis multiple sclerosis because this was such a severe disease. Now with Rebif, the status was lifted and it became a much more manageable disease, as Professor Chan already said. We created all sorts of devices for that because that is something Serono has traditionally quite strong in, and one of them is really with this, where you can basically have an electronic injection device and look in your smartphone whether you've taken the drug successfully and so on. This is the current production site. It's in Switzerland, in Vevey, and it's one of the largest and most technologically advanced biotech manufacturing sites in the world. We are even expanding that further, all due to the success of Rebirth. Without that wouldn't be possible. And this is, I think, Michel, the number that you should be particularly proud of. I mean, 500,000 patients and counting are today benefiting from Rebirth. I believe that is really something. I don't think a man can be more proud of an achievement than that. I find that fascinating, that number. And it is inspiring not only to the patients and the medical doctors who prescribe this drug and the families who have hopes. It's also inspiring for the scientists who are still working on multiple sclerosis because this really shows you can beat that disease. And I would like to briefly tell you a little bit about what we are currently doing. This will be very brief, I promise. This is DNA, as it is wrapped around the histone twice. And you can see here that one of those DNA strands actually stops suddenly here. This is because we invented a drug called cladribine, which is 2-chloroadenosine, and that actually prevents this other strand actually from growing, and that can be very deadly, in particular for B cells and T cells, both cell types which are involved in the genealogy of multiple sclerosis. And you can actually test that in the clinics, and you can see a massive reduction of cumulative incidence in clinically definite MS. So what the nice thing is about cladribine is actually its posology. You give this tablet; it's an oral treatment, a pill. You take this on 10 days. Consecutively on year one, and you have protection for a full year. Then you take it in a second year, again, only for 10 days, and then you have protection for 3 consecutive years. That is something where we are getting close to that patients can actually start forgetting about the disease, which is something they want. So it is a nice drug. We've just filed that or got approval from the EMEA. For this material, and in the FDA, the approval is still pending. We just filed that, and we received an acceptance letter of the filing from the FDA. Now there's one other drug I wanted to briefly show you that's very, very young, but again showing how much Michel's work actually is inspiring us. This is a kinase. It's called Bouton's tyrosine kinase. It's a kinase mainly expressed in B cells. And we developed an inhibitor of that, which is actually pretty difficult. This is the site where you would want to get in, but any molecule you design there would slip out very quickly because it's very shallow as a binding site. So we used a little trick. We created a covalent bond between the drug and the enzyme to actually get it in there. The compound is called evobrutinib, and that compound, we just announced earlier this year that our evobrutinib phase 2 study met its primary endpoint. We are currently designing the phase 3 study, and we hope that this could actually bring further benefit to patients. So research never stops, obviously, but these compounds will probably never be in competition with Rebif because this will be more for severe cases. and Rebif, I think, is something else. So this is the man behind the drug. I like that photograph so much. I believe it's what you see is really in the foreground, a model of interferon-beta cast in metal. And yeah, it stands at Kadimastem, and I'm told that is a very flourishing startup company that looks into the advancement of stem cells as the next generation of therapies. And Michel told me yesterday evening that his people are working very, very hard, getting up very much, actually in the night, and at 2 o'clock they have to prepare some injections and some syringes. Quite amazing, and it must be very inspiring to work for Michel, I guess. So. We also, there's another anniversary that you might have witnessed, and actually it's the anniversary of Rebif itself. It got market approval 20 years ago, and we celebrated that in Munich. And actually there was it was a little bit unfortunate that Michel couldn't really join us. But as Michel and us, we actually, when we think together, we always come up with a pragmatic and innovative solution, we believe. So in this particular case, we decided to, okay, if you have to be in Israel, then we will beam you to Munich, and you will then electronically answer to the questions of the interviewers. And that's what you can see here. We had a 3-dimensional hologram of Michel. He was standing there on stage and answering the questions of the interviewer. It was very well received, I can tell. And this little thing here, this is not a halo of Michel. It's an artifact of the photograph of my iPhone. Right. It is the 80th birthday, and I think we are obviously there should be a present, and we thought that we bring you something that I had difficulties to bring into this room. It's 120 trees that we are going to plant in the Chaim Weizmann forest near Jerusalem, and we would present that to Professor Michel Revel in honor of his extraordinary scientific achievements in the field of biotechnology in general and in the creation of recombinant interferon-beta in particular, which enabled the leap forward in the treatment of multiple sclerosis patients worldwide. Michel, happy birthday, many happy returns.